Welcome to today's lesson on protein synthesis. Before we begin, let's take a look at this picture because it really frames this whole unit well. In our cells are nuclei, and the nuclei contain 46 chromosomes in a human cell. Those chromosomes are made up of DNA, and the DNA has specific segments called genes, and these are what we inherit from our parents that make us who we are. Now the genes get copied through a process known as transcription, through which we create a modified kind of copy called RNA. And that RNA leaves the nucleus and is taken to the ribosome, which is the protein factory, and that mRNA is decoded or translated through a process known as translation and used to make protein. And it's proteins that actually express the physical traits that we have. So proteins are responsible for traits like eye color, for example. All right, so protein synthesis is the production of polypeptide chains or proteins, and as we mentioned in the previous slide, it involves transcription and translation. Transcription is how mRNA is made, and through transcription, we're going to take the gene region in one strand of DNA and use it as a template to make an mRNA strand. And that green is the mRNA strand right here. And this happens, so we go from DNA to RNA through the process known as transcription. Then that mRNA is taken to the cytoplasm, to the ribosome, where it's deciphered and translated into a protein. So this process of transcription is how mRNA is made, and it takes place in the nucleus. And the process of taking the mRNA and translating it into protein is called translation, and this takes place in the cytoplasm in the ribosomes. Okay, transcription is a pretty simple process, and again, this is how mRNA is made. So the DNA is going to unwind and unzip into two strands. One strand of the DNA is going to be used as a template to build an mRNA strand. This over here is the mRNA strand. And the enzyme that's going to help with this process is RNA polymerase. It is going to bring in the RNA nucleotides and assemble them in the correct order using the base pairing rules. So if it encounters a G, RNA polymerase is going to put in a C. If it encounters a G, it's going to put in a C again. If it encounters an A, it's going to put in a U because if you remember, RNA doesn't have any T's. Instead of thymine, RNA is going to use uracil. Whenever it encounters a T, it's going to add in an A, and it's going to keep building this strand until the entire mRNA strand has been made. Once the mRNA strand is complete, it's going to actually leave the nucleus through the nuclear pores. This is what that looks like. So after transcription is done, you can see the mRNA is complete and it is leaving through the nuclear pores and entering the cytoplasm. This mRNA contains the message that's inside of the gene, and that message needs to be deciphered and translated into a protein, which is what's going to happen next. Through translation, that mRNA is going to be decoded and translated into a protein. So the first thing that happens in the process of translation is the mRNA has just left the nucleus through the nuclear pore, and initiation is going to take place. Initiation means that the ribosome and the tRNA are going to attach to the mRNA strand. So the ribosome is actually made up of two subunits, a large subunit and a small subunit, and they're going to basically hook onto the mRNA strand. And then this structure over here, if you remember, is called tRNA, and the tRNA is short for transfer RNA, and it's bringing in the amino acids. The tRNA is used to carry amino acids. So the first tRNA is going to help, and it's going to attach to the mRNA. So during initiation, the ribosome attaches to the mRNA strand, and the tRNA, the very first tRNA, is going to attach to the mRNA strand. If you remember, the start codon on the mRNA is always AUG. That is the start codon. So the first tRNA that's going to bond with AUG is going to have the anticodon UAC. If you remember, the tRNA has anticodons, the mRNA has codons, and they are complementary. So A goes with U goes with A, and G goes with C. Now AUG, the start codon, codes for the amino acid methionine, and we can double check that. So A UG. It codes for methionine, which is our very first amino acid. There is methionine attached to the transfer RNA. So that's the first thing that happens. This is called initiation, and it's the start of translation. What's going to happen next is a new tRNA is going to come, and it's going to bind to the next codon. So the next codon is, so first was AUG, the next codon is UGU. So the next tRNA that comes in has the anticodon ACA, which is complementary to UGU. And the second tRNA is carrying a second amino acid. So this process is actually going to continue, and it's called elongation. New tRNA are going to keep coming in, and we're going to be adding in amino acids. By the way, between the amino acids, peptide bonds will form that will hold this chain together. This process keeps going, so we keep reading the mRNA and bringing in new tRNAs until we reach a stop codon. If you remember, the three stop codons are UAA, UAG, and UGA. Once we hit one of those, the process of translation is over and the amino acid chain is released and it's free to go and do its job. The ribosome is going to detach and the tRNA is going to detach. It's all going to fall apart and the polypeptide chain will be free and the translation process will be complete. So here's another picture showing you this. We have our mRNA strand. It's being read one codon at a time, 3 letters at a time. The transfer RNAs are coming in. They each have the anticodon at the end, which is complementary to the codon. And each transfer RNA is bringing in an amino acid. And the amino acid chain is growing here. Peptide bonds are forming between them. And this keeps happening until the stop codon is reached. Okay, so one more summary slide and we're finished. There's our DNA. And we started with the DNA, we started with the gene, and through the process of transcription, an mRNA strand was made, which is right here, right? So we took one of the strands of DNA, used it as a template, and we built the mRNA through the process of transcription. And then that mRNA was able to leave the nucleus and attach to the ribosome, and it was translated 3 letters at a time into an amino acid through the process of translation. So I hope that was helpful. Thank you so much for listening.